Okay. So we left off looking at angiosperms. So we have the different life cycles essentially of how do they get their embryo sacs or their megagametophyte established. So I only want you to know the polygonum type from start to finish, which is short because you just have that megaspore mother cell or megasporocyte going through meiosis. And you end up with 4 cells from that. And then one dies off, and then that last remaining one goes through 3 rounds of mitosis. And then you have some migration of the cells in the embryo sac or the megagametophyte. And you end up with 8 nuclei, 7 cells. So there's 2 nuclei stuffed into that so-called central cell. And that's important because that's going to become the endosperm when one of the sperm fertilizes that or merges with that. And then we briefly touched on, yes, there's other options. 30% of angiosperms do other things in order to get their megagametophyte developed. We looked more seriously at the lilium because that's what you'll look at in lab. Hey, how did the soap making go? You did that last week, right?
Well,
good.
Yeah. Smelled great.
Yeah, didn't it? I love walking into that lab when
It's a lot now than the Orgo one that I did 2 days before.
see how useful plants are. You can make soap, your own homemade
Right.
soap. So I saw a question. Did you have a question? Going back to this idea? Sorry.
Do you want us to know that range or?
For which one?
For the female gametophyte, can have 4 to 16 nuclei. That's
No.
option. Just, okay.
I'm just showing you some options. It's, and one thing though, with the polygonum type,
Mhm.
what I do want you to catch is that with these 2 polar nuclei in one cell, when a sperm comes here and fuses with that cell and the 2 polar nuclei, then you're up to 3 nuclei. So I do want you to catch that. In the polygonum type, you end up with 3N. And that's what's going to create the endosperm. So the endosperm tissue is actually 3N. When you crack open a seed and look at endosperm in polygonum type, it's 3N. So that's kind of weird. So I do want you to catch that idea. So we looked at that idea of double fertilization. So if everything goes okay, you're going to have 2 sperm getting discharged in this area with, through the pollen tube, and then one of the sperm will fertilize the egg, and that's going to become the baby sporophyte, or the developing embryo, which turns into the baby sporophyte. These 2 polar nuclei, and that second sperm fuse, and then you're up to the triploid state, the 3N. So that's the double fertilization idea. Both sperm do something. We didn't see that in the angiosperm. All right. One of these sperm just kind of died off. So, okay, then I think, so that's review of what we talked about before. And then I wanted to get into the microgametophyte, which is looking at the pollen grains now in the angiosperm. So they're a little bit more straightforward than with the development of the megagametophyte. So here, we're gonna go to the male part of the flower, which is the stamen. If you take all the male parts together, you call it the stamen. And we're gonna zoom into the anther. So that is where you see the pollen showing up. So really, the anther is microsporangium tissue. Remember, sporangium tissue, if you look inside the sporangium area, you're gonna see spores. With microsporangium, they're making the pollen grains in this case. And it's usually where you see 4, they call them pollen chambers, 4 of them, 4 pollen chambers. So look for that pattern when you're looking at it under the microscope. And you see under the microscope something like this. So this is a perfect flower taken in cross section. And so you have to imagine, all of this swooping around all the way. in a continuous ring around the central part. So the central part, that's the female. Okay, that's the female part of the flower. So you're looking at the ovary and it looks like 6 ovules inside. When you look there, so there's 6 ovules. And then the anthers are, surrounding the female. And so that's a complete one. And then you have to imagine it going all the way around in completion. And so these are the so-called pollen chambers. That's where the pollen is going to be developing. So with the pollen and getting that going, it's fairly straightforward. What we see happening is a microspore mother cell or microsporocyte is going to be in those pollen chambers. And you're going to get meiosis again. And if you have one cell going through meiosis, you end up with 4. All of those survive. All of them develop into pollen grains. A little simpler than with gymnosperms. The pollen grain is only 2 cells when it gets shed, or when pollination occurs, let's put it that way. When pollination occurs, it's only 2 cells. You have generative cell and tube cell. And nowhere there is it saying anything about sperm, okay? So in this state right here with the generative cell and the tube cell, that's how it is when pollination occurs. So when the pollen gets to the stigma of the flower, there's no sperm yet.
So each one of those have, like a.
Yes. Yep. So eventually you're going to see the generative cell, it's going to go through one more division to give you the 2 sperm, okay? But it is shed in, or I guess I should say pollination occurs when there's only 2 cells. And that's different with the gymnosperms. We said when pollen is shed with the gymnosperms, there's 4 cells. Still no sperm, but it's 4 cells. So the angiosperms have simplified. They're like, let's just do 2 and let's get pollination to occur, and then they'll kick into producing the sperm. Okay, so I put this is back to the gymnosperms. So it's different. We had 4 cells back then, the gymnosperms, and it's a little bit more complicated. So the angiosperm simplified this and just said, let's do 2 cells and get it going that way. So once you have that microspore mother cell doing all of that, going through meiosis, 4 cells, each cell becomes pollen grain, then you would see this in the anther areas. And so now you can see that those look more like pollen grains showing up in each pollen chamber. Okay. So now pollination would be a good idea to happen at this point. And pollen, here's a cross section with pollen grains where you can see that they're quite ornate. You see the 4 pollen chambers. Hard with my red to show up on this, but these are the pollen chambers. And you can see how ornate the pollen looks, pollen grains. So I'll talk a little bit more about that. So this is just another idea that you're going to get that tube cell that's going to grow out and help deliver the sperm that eventually form after the generative cell does 1 round of cell division. Okay, so you really, when you look at a pollen grain, that's your microgametophyte essentially. Okay, because that those microspores developed into the gametophyte, which is the pollen grain in this case. All right, so you have the generative cell, the tube cell, and then here they're trying to point out a little bit more about the design of pollen grains. And so the outer portion is called the exine area of the pollen grain. And that's very specialized proteins that make up that coat, so to speak. And then we tend to see so-called apertures that are present on the pollen grains, and we'll talk more about that. That's usually where the pollen tube grows or germinates out of. It's an easier exit area out of the pollen grain for the tube cell to start growing out of. So no sperm yet when you have pollination occurring. It's still considered immature pollen grains. So you can, let's see, was it last winter, some students made some pollen grain slides from a variety of different flowers that so they collected pollen through the summer. I guess it was last year sometime. And then so they collected pollen grains over the summer and then they made slides. And so I'm going to pull those out. So in lab this week, you can take a peek at different pollen grains. So they can be fairly simple. And here's the aperture right there. So that's where the tube cell will grow out of. You see one aperture. Here you see there's really 3 apertures, little different design, but there's 1, 2, and then there's a third one back here that is only partially visible. But again, you can see the exine portion is very unique. Here's another one.
A fingerprint.
Yeah, it is absolutely a fingerprint idea. And so here again, this is actually 3 apertures. There's one there, and then the last one that you can't see. It's off the image there. You can't see it. Very beautiful. They can be, but they're all unique. People make artwork out of pollen grains. So, I mean, look at that diversity. It's amazing. And really, as far as we can tell, every plant species has unique exine area of the pollen grain. And they use that. I mean, they've been able to use that to solve murders in forensic, what is that? Forensic science or whatever, where the case study I read, a woman was murdered at a location outdoors, and then her body was moved, and so they found her body. They found pollen on her, tracked that pollen. Fortunately for solving the case, they were able to find out that patch of woods where that pollen was uniquely there in that area, the geographic area. So they pinpointed the location of the spot she was murdered. And then after a lot of investigative work, they had a suspect. And when they tested the back of the bed of his pickup, they found those pollen grains. So they linked together that he moved her body and put her body in the pickup truck. And they were able to use that as testimony and solve, the murder. So, I mean, that's a pretty unusual example of botany cases of, how to use plants in the real world, but a very powerful mechanism there. So with the idea here that pollen grains are so unique, it can be of use. So I'm going to pull out some of the pollen grain slides, and you can look at those in lab this week. I thought that was a really pretty image caught on the spider web. I wonder if spiders eat pollen. Does anyone know? I don't know. Yeah. Can deal with that.
They use it to attract predators.
Yeah.
Prey, rather.
Maybe, hey, if it catches a prey eye, like, ooh, what's that? Something shiny. This is a fly eye. Isn't that bizarre? Pollen grains on a fly eye. That's amazing. Yeah, probably. Yeah, get that off. Wonder if they get allergies to pollen. Do flies sneeze? Oh, yeah, true. So, okay, so putting it together here, you have pollination occurring in this drawing where they have pollen grains getting transferred. In this case, it's a perfect flower that they're showing, where the anther containing the pollen grains are going right to the female part of the same flower. And that can happen with some species, but many species prefer to get pollen from another nearby plant that has the pollen for that species. So, but for the ease of showing this, let's go with this. And so they're showing it looks like 3 pollen grains landing on the stigma of the female part of the flower. And then you see the pollen tubes growing down. And their job, the pollen tube's job is to get the sperm to the egg. And so you're seeing it, you know, with the micropyle area, the egg is right there. And so that's convenient. Once they get there, they're able to get the sperm to the megagametophyte or the embryo sac, and then both sperm participate. One fertilizes the egg, the other fuses with the polar nuclei. And so this is, yeah, I guess it really is showing both pollination and double fertilization. Okay, I'm surprised they didn't put double fertilization. It's kind of a misnomer because they're not really fertilizing 2 eggs, so it's a little confusing. So when they say double fertilization, it just remember both egg participates, both sperm participates, sorry. One sperm goes to the egg, one sperm fuses with the polar nuclei, and then that 3 nuclei then in here will become the endosperm. So pollination, we're going to talk about that because we have to think about different ways that angiosperms evolved to allow for efficient pollination. They don't really rely on wind so much. Most species rely on some kind of pollinator organism. But here is the lovely dandelion. And you can see how well the pollen is sticking to the stigma. Really beautiful image there. So this is the life cycle in total for the angiosperms. This is a polygonum type, okay, where they're just showing with the female development, it's just the polygonum type, right? So that's the only one you need to know. Any detail and looking at what happens when you reach the completion of it with the embryo sac and the rearrangement of the cells there. And then they also have on this image the progression of the microspores developing into the pollen grain that just have 2 cells initially, and then eventually the sperm show up. So once you have that egg fertilized, we still say it's a zygote. And then of course the zygote has to go through a lot of growth, lots of mitosis, differentiation. And then we start calling it the embryo. And then eventually it's really the baby sporophyte, because when that seed gets dispersed and starts to germinate and grow, you're back to the sporophyte phase. Okay, when you see that grow. Questions on the life cycle? So this is the one from your book. These are just, if you want to kind of zoom in and look at it when you're studying. And then this is not from your book, but if this helps you while you're studying.
So overall, is like the sport, like the flower?
The plant, the whole plant, yeah, the whole thing. We tend to zoom in on the flower.
Because that's where.
Yeah, yeah. And so it's the flower is very specialized to allow for, okay, we got to get the sperm eventually to show up. We got to get the eggs going, and they happen to do it. You know, angiosperms do it with the formation of a flower. Gymnosperms do it with cones. So, but the whole plant that you look at, the whole dandelion is the sporophyte. So this is saying the same thing. It's just maybe a little bit more words. It's a little clearer to see. So you can use this as well for studying. And they actually put on here double fertilization. Here, and they're showing the 3N with the endosperm. So that that polar nuclei and the sperm and all fuse, you get 3N. So that's when now that has to go through. You know, we kind of focus in on the zygote. Certainly, that's going to go through a lot of growth and differentiation to get the embryo and then the baby sporophyte inside the seed, but that endosperm also has to go through a lot of growth, just mitosis, so it can develop into the endosperm tissue, which is going to help feed that baby initially until the plant can start doing enough photosynthesis. So here again, just another slide to get you, if you want to study off of this, that's fine too. So there's just some options for you to look at as you're piecing this all together. So here, what they're trying to show is the double fertilization has already happened. So this is your zygote. The sperm already fertilized the egg, and that has to go through a lot of growth to become the embryo and the baby sporophyte. And then they're showing that the endosperm is developing as well. And that's a different level of how many nuclei. It's triploid instead of diploid. And so the endosperm is really important, you know, so now they're saying, okay, when things are pretty much ready to go and that seed's ready, you typically have some nice endosperm tissue that's triploid. You have the embryo that's diploid. And then remember the integument, again, just like with the gymnosperms, the integument that's really surrounding and protecting, that's going to become your seed coat. So lots of growth and differentiation needed to get everything to work out, so you get a viable seed. And of course, depending on what type of angiosperm you have, the seeds will be a little different. So we talked a little bit about that way back during week 1 when you thought about seeds and got some seeds germinating in lab. And so we'll talk a little bit more about this, because when you look at seed development, you need to think about, well, you can often tell if it's going to be an angiosperm or a monocot or a dicot, just by dissecting the seed and looking at it. So we'll get into that. Oh, one other thing. Do they have it on this? Okay, so on this one, I just wanted to point out that you have, like, let's look at this seed on the top. You have both endosperm showing up in that seed and cotyledons. And they're both pretty much serving the same function. They're feeding the baby. So as that baby sporophyte, the seed germinates and starts growing and the baby sporophyte emerges, but it's not ready to do photosynthesis to full tilt. So then the endosperm and the cotyledons help provide nutrition. So some seeds are like that. Some seeds are more like what you see on the bottom here, where you see the way the artist drew this, there's no endosperm. It's not there. So what happened? What they tend to see is that the endosperm kind of, the way I think about it is like the cotyledons kind of suck away all the endosperm nutrition, and they're the ones that are like, I'm going to feed you, and not the endosperm, the cotyledons. So basically what they see during seed development is it starts out with an endosperm, but then the cotyledons kind of surpass what's going on and really get beefed up. And so you, like with bean seeds, most of what you are eating is really cotyledon tissue. And so, but it's the same idea, whether you're talking about cotyledons or endosperm, they're helping feed the baby. It's nutrition for the baby sporophyte. And then with grasses, it's corn, you get, I'm not going to ask you to know in lecture, I'm not going to ask you to know any of the details of that. But of course, basic anatomy of the seed coat, what are cotyledons, what are endosperm, what's endosperm, those ideas are important. Yes.
Polio, right? So just like a root.
Yeah.
Emerge.
Yeah.
Okay.
So anytime you see RHIZA, yep, you should think about roots. So then here they're looking at, okay, let's say that seed is viable, it gets dispersed, it starts to germinate and grow. Then you can see kind of the progression here. This one, the cotyledon has fully taken over. There's no endosperm. But you can see in this case, the cotyledons end up above the soil. And that's not always the case. Sometimes they stay below. It just depends on how the stem tissue grows in relationship to where the cotyledons are. So sometimes you'll see that with beans, for example, you'll see that. If any of you grew like kidney beans in the lab, you saw the cotyledons. So they look different. They're not considered the true leaves because they're kind of big and plump. They don't really look. And so that's why they're saying first true leaves are finally showing up, and then as the leaves take over doing photosynthesis and taking care of making sure there's enough nutrition and the nutrition has been fully kind of siphoned out of the cotyledons usually die and drop off, which is normal. So here you're looking at a couple of ideas where the bean plant, you see the cotyledon above ground, but the pea plant, B, here, the cotyledon stays below ground. So sometimes you just don't see the cotyledon. And then the corn, you can see the coleoptile is just, I don't want to consider it like a sheath that goes over the first true leaves that will come out. So with the seed development too, we talked about pollen grain being very ornate. Sometimes you can see very ornate seeds as well, especially when you look really close up. So a lot of these images are taken from competitions where they submit their photos for competition. Yes.
What makes the seed not viable?
Viable?
Yeah.
If something went wrong during the development of the embryo, where it just isn't gonna make it, there's that idea. Because if you buy a packet of seeds, they often say like germination rate 80%. And that usually means, in our batch back at the company, when we tried to germinate these seeds, not all of them made it. And it's that's a complicated answer to your question. It's a really good question, but it's, a lot of it is just something happened to the sporophyte. And then if you don't give the seed everything it needs to germinate is the other thing. So user error on, when you buy those seeds and you start working with them, if you don't know how to work with them, that can often make the germination rate really low.
The seed coats can always be kind of thick, so.
Yeah, and like sometimes they'll say, yeah, exactly. They'll say, scar, like take a sharp knife and actually scratch the seed coat. That's usually what they tell like regular people to do. Like you could also use enzymes on it, but most people don't like, I'm gonna buy some enzymes and so, yeah.
I have another following question, but do the male plants produce the seeds or the female?
The female, yeah.
How about like in cannabis, when the male plants generate the seeds and the females more so the flower? Or the flowers and.
Yeah, the flower
Flowers can have either one, or they can have both.
yeah, so with cannabis, they usually, the male flowers, it's the job of delivering pollen. Female flowers, that's where you're gonna get the seed development will show up. Yeah. So the female part of the plant, that's where you're gonna find the egg, and you gotta get the egg to get fertilized to eventually end up with a seed. And so if you can follow that progression through, you can always figure out, who's doing what when it comes to the different flower types. So yeah, we've been focusing on the idea that I've been showing you images of a perfect flower that has both male and female structures. But we're going to learn that there's plenty of angiosperms that have imperfect flowers where they're just a male flower or a separate female flower. Okay, so I'm going to switch into the next slide set, which should be in EDUCAD as part 2. And wanted to talk a little bit. Okay, so I wanted to talk a little bit about angiosperms and their evolution and the fact that they're incredibly successful. They're the most dominant plant on Earth. And so one of the reasons for that is the idea that they have partnered up with pollinators in order to get successful pollination to occur. Whereas when you think about the other seed plants, the gymnosperms, they rely most heavily on wind, just wind currents, air currents. When you look at more primitive plants early on, when they were first starting out, insects like beetles, crawling insects were often what they see as pollinators. and hypothesized that was a pretty common strategy way back millions of years ago. And of course, once the flying insects came along, once they evolved, then coevolution with those organisms also started to occur with the angiosperms at that time. So you can get nice pollination, and you can get further distances. Once you have some organism that can fly, they can fly further than, a crawling insect. They can go further than a crawling insect. So we're going to think about, what is a plant doing to try to promote this partnership? And so you have to think about it from the pollinator's perspective, like what is going to, and then also think about it like marketing. Like if you're going to put on an advertising campaign, at least when you're trying to catch a human's eye, you usually try to use bright contrasting colors. Maybe, by the way, like I've noticed, have you noticed, maybe you haven't, but I've noticed this year more than ever, when I'm walking around in the buildings, there's a definite scent in the air.
Yeah.
Have you noticed that? So I'm thinking they must be doing that on purpose, because they didn't do that last year. What made me think of that is scent is a big attractant, right? And so if you can pick up on I like that smell. And then you can target, where's that coming from? And so from the flowers, plant's perspective, smell, nectar or odor is a good strategy. Reward the pollinator, give some nectar, a nice sugary treat. That's a really good mechanism. And then if everything goes okay and pollination happens, then hopefully the seeds develop. And then after that, we have to talk about, well, how do they disperse the seeds? Because that's another thing that's really important for the angiosperm. So we're going to focus a little bit more on pollination before we get to seeds. But let's think about, this progression of the angiosperms through evolutionary time. There's a lot of reasons besides the fact that they partnered with animals and for pollination and seed dispersal. You do have some amazing adaptations. Believe it or not, most of those leaves are pretty tough. There's a lot of, nice cell wall tissue. They can withstand the weather and environment. You have the vascular tissue was also of a huge adaptation over time that was very successful. And angiosperms got more sophisticated in the sense that now with angiosperms, we see vessels showing up. So now when you think about angiosperms, vessels were great because they're wider in diameter to allow for faster water and mineral movement through the body. So most angiosperms, they have both vessels and tracheids. Tracheids are the more primitive type, but don't forget, angiosperms also have those. But when you're thinking about, what made them so successful, this list is, the idea that they came up with vessels that made them more successful in terms of being able to spread and adapt to more areas than the gymnosperms. Phloem, they also evolved sieve tube elements and companion cells, whereas the gymnosperms just have sieve cells, which are a little bit more primitive. And so that advancement, that evolutionary adaptation is another reason they point to. This is why angiosperms are so successful. The seed coat usually is really tough and protective. They think being able to drop your leaves and kind of go dormant over winter is actually a good adaptation for angiosperms. It's a really good way to handle cold weather. I'll just go to sleep until spring, essentially, so that's a good adaptation. And then like the partnership with pollination and seed dispersal. And then of course, angiosperms, as we know, they make, all plants, but especially angiosperms have wonderful chemical defenses and ability to protect themselves that way from getting eaten by or attacked by different organisms. So this is kind of showing some ideas of how they've been tracking what's going on with the evolution of the angiosperms. And you're seeing some of the little bit more detail of more ancient. So the basal angiosperms, that means more primitive type. And then here you're seeing, we're going to talk about monocots and compare monocots to, we tend to shorten it and just say dicots, but technically eudicots is the correct way to say it. But here you can see that you have a difference in number of cotyledons. Talk a little bit more about that. Or, no, sorry, what I meant to say is, we're gonna get in, well, yeah, cotyledons and apertures, we're gonna get into that. So, here, basal angiosperms is the leftmost column. The water lily is probably maybe what more familiar to you. So they're showing, like what do their flowers look like? So the different rows. And then pollen, so when they say opening, they're referring to the aperture. So when you look at one opening versus three openings. So if you look here, as I'm going to be asking you especially to compare monocots and dicots. So these last two columns in particular are important. And then I'm going to talk a little bit about the evolution of the flower. And so that flower row is going to be important. Okay. But you can see that you have the aperture of the pollen grain can vary depending on if you're looking at these different groups of plants. So the monocots have one aperture opening, the dicots have three apertures or openings. And then you can see there's difference in the amount of cotyledons inside the seed, one or two. Leaf vein, we've gotten into that a little bit already when we looked at leaf anatomy, where we have parallel versus net-like vein pattern between the monocot and dicot. And then we've already talked about, so some of this is just review. We've already talked about the idea of how do they arrange their vascular tissue. Monocots in their stem, they always have the scattered vascular tissue, whereas the dicots have a ring of tissue. And then root system, I don't know if we got too much into this, but with the root systems, you tend to see mostly fibrous roots with monocots. It's not 100%. And you tend to see mostly a taproot system in dicots, but again, that can vary. So this is a nice kind of one-stop shopping here. You can kind of compare what's going on. But if we look at the dicots, you tend to see that idea of two cotyledons. I'll be asking about, so this slide is important in the sense that we're going to talk about flower design too. So when you look at dicot flowers, they tend to have their flower parts in multiples of 4 or 5. So they might have 4. The easiest thing to do is look at the flower petals. So if you look at the flower petals, there's 5 flower petals. If you look at the, here you can see the pollen grain. So those are the anthers. 1, 2, 3, 4, 5. And then that's the stigma right there. So you have 5 anthers and filaments. You have 5 petals. So that's a tip off. You're looking at a dicot species. Very good. Yes, yes. And orchids too. Yeah, so you got to be careful about that. Yep. So look at dicots win. 80% of all angiosperms are dicots. That's a good thing to remember. Especially if you're out trying to figure out what are you looking at when you see a strange plant. Chances are it's a dicot. You have an 80% chance of that. So woody trees and shrubs, lots of herbaceous species fit into the dicot. Netted veination, 3 aperture or openings.
Trying to use one of those like plant identifier apps or like Limbos that can narrow it down to as a dicot. So I guess that.
Yeah, with a little bit of training. Yeah, I know. But you have to realize so many people know nothing about plants, so hopefully those apps will get a little bit more sophisticated. Monocots, you tend to look for groups of 3 petals, 3 anthers. What am I? So I tend to zoom in, especially if it's, got if it's a male flower, a perfect flower, I tend to look at, the anthers are usually easy to spot.
Yeah, trillium, right?
Yeah. So about 20% of angiosperms are monocots, roughly. So here's a comparison side by side of the monocot on the left, dicot on the right. Here's another comparison slide. This is nice. So this is showing pretty much what's on that table, that black and white table, except now they have some nice visuals. So this is, I think, a little easier to maybe use as a study tool. Okay, yeah, I like that. That's nice. And then this is just another one I found that a little sillier, but does the same idea. Visuals are always good instead of just words. Huh? Amoeba Sisters?
I recognize that style.
Oh, yeah, it says it on the bottom corner there too.
Yeah, Amoeba Sisters.
No,
Good eye.
They make some fun science memes that my mom shared.
Yeah.
Science teacher, so.
Yeah, it helps. So I'm guessing she uses a lot of memes too, because it can really help. Anything that helps you to get it to stick. All right, so let's think about flower design in the evolutionary timeline. So when they are looking at the more ancient plants that, you know, evolved first after the gymnosperms, and we start to see flowers showing up, you tend to see this idea of lots of parts. They started out kind of flamboyant and extravagant and went with, let's make lots of flower petals and lots of, you know, stamens. And so, I mean, look at that. That's just extreme. And then you have the female portion too, extreme, just lots. And then you tend to see the flower tends to have a longer overall axis, like it's a taller flower. Lots of parts that aren't fused together. So all these stamen, you know, they're just separate from each other. They're clustered, but they're definite separate. And the ovary sits on top of all the other flower parts. It's a superior ovary. Those are very primitive. Also, this idea of radially symmetrical. So this slide's really important as you're studying, because I'm going to be asking about what's a primitive feature, what's an advanced feature. And so radially symmetrical means if you look down on the flower and you imagine cutting it in half, you can cut it any which way and you're going to get mirror images of the two halves. So if you can imagine cutting that, doesn't really matter how you cut it. If you compare that to the image on the bottom here, where this is an example, this is an orchid. Orchids are very evolutionarily advanced. You can see that they've pared down how many flower parts they have. They've simplified. And so you have fewer petals, fewer male-female structures. You tend to get fusion of the structures in the flower. And bilaterally symmetrical means if you look at the flower, like you can only cut it from top to bottom in the way they have this picture in order to get mirror images. That you have to have mirror images. So that's bilateral. We're bilaterally symmetrical. There's only one way to cut yourself in half. So then you get the inferior ovary idea, where they tuck away the ovary and are a little bit more protective, since that's such a precious area for the plant. Okay, so yeah, I know. I mean, it's just like, here I am. So this is a nice one. So magnolias, the magnolia tree, that's a really good example of an ancient primitive plant. Their flowers are beautiful. I love their flowers. I love the tree design too. There's some magnolia trees people have landscaped with through town, and it's just kind of cool. The branches look really cool. I gotta get one for my yard. So, but you can see lots of stamens, lots of pistils, so the male and female parts, lots of petals, superior ovary, and then compare that to an orchid where they sunk the ovary, they infused parts together, they have fewer petals. Okay, so here you see the stamen and pistil are fused together. So that's a really good example. And then this, just remember the idea of where is the ovary with respect to the rest of the flower tissue. And over time, we do see this idea in the fossil record and through other studies that go from the early primitive angiosperms having a superior, and then they get more advanced and go semi-inferior, and then the most advanced are full inferior. And then here's the idea of radially or bilaterally symmetrical. Okay. So, of course, there's all kinds of debate. How did the flower evolve? Most people think it's from leaf tissue, and it just became specialized into the flower. Some say, well, what about stems, maybe? But I think that's kind of gone away. I think there's more, with more data. I think most of them are saying, yeah, it looks more like modified leaves. So the leaf, the flower petals themselves, they think might have developed from the stamen tissue that formed, and then slowly over time, they kind of specialize into becoming the petals. So remember, if you look at a perfect flower, the male portions of the flower are the closest to the petals. And so it kind of makes sense that they think the stamens, over time, some of the stamens became flower petals. And then petal fusion, you see in more advanced, evolutionarily advanced plants, you tend to see more of a, they call it a tubular corolla. And that's just what the shape it takes when you have a bunch of the petals fused. You get that tube-like look. And then often you get fusion of the stamens. So let me just end on this, where you can see a leaf surface that started to devote itself to reproduction. So you can see here with the development of the pollen, the anther over time. So it's a really cool progression. And then with the female, they're showing the megasporangium tissue slowly evolving to have that defined pistil shape that we talk about. Okay, all right, thank you for your patience. We'll stop there and continue on with flower design on Wednesday.